Hello, I'm Saumya Kumar and I did my PhD in the laboratory of Luís Graça at Instituto de Medicina Molecular. The importance of antibodies in protection against infection had never been as significantly realized as during the current COVID-19 pandemic. It is critical to understand the regulation of antibody responses to design better vaccines against many infectious diseases. The protective nature of these antibodies relies on a tightly regulated process that takes place in special microstructures called germinal centers within the secondary lymphoid organs. The efficient antibody response relies on a subset of CD4 positive T cells called T follicular helper or TFH cells. The TFH cells help B cells to produce high affinity protective antibodies within the germinal centers. Another subset of CD4 T cells called T follicular regulatory or TFR cells regulates germinal center responses. Both of these cell subsets are important for efficient antibody immune responses against pathogens. However, studying these cell subsets in humans has been very difficult. Most studies about these cells in humans use blood cells, as access to secondary lymphoid organs where these cells are acting is difficult. Tonsils resulting from tonsillectomies have been used to study TFH and TFR cells, but their phenotype is very different from blood. One aim of my project was to understand these differences of TFH and TFR cells observed across different human tissues. For this purpose, we generated single-cell transcriptomics datasets of TFH and TFR cells from 3 different lymphoid tissues in humans: blood, tonsils, and lymph nodes. We investigated the relationship of TFH cells from the 3 tissues using a pseudo-time algorithm, a method that arranges the cells according to their changing transcriptomics profile. We found that across this pseudo-time axis, the TFH cells were arranged in an order of increasing maturity, where the most mature cells from the tonsil laid on one side of the axis, while the most immature cells from the blood were on the other end of the axis. In this way, we were able to generate a timeline of changing gene expression as TFH cells mature. We had hypothesized the maturation of TFR cells showed a bifurcated developmental path originating from Treg cells. Therefore, using similar methods as above, we were able to show that TFR cell differentiation follows a bifurcated trajectory, where blood TFR cells originating from Tregs forms one arm and are immature cells, while tissue TFR cells is on the other arm and show a mature TFR phenotype. A second part of my thesis focused on understanding the different TFH subsets that arise with distinct types of immunization. In this project, TFH cells from secondary lymphoid organs were collected from mice immunized with either type 1 or type 2 adjuvants. We generated single-cell transcriptomics of these TFH cells, and we were able to identify the different TFH subsets and dissect the heterogeneity of these subsets within each immunization. In conclusion, the findings from both projects of my thesis showed the advantage in using a combination approach, where big data generated from controlled experiments was analyzed using computational methods to answer the questions which were otherwise difficult to approach without such combination of in vivo experiments and computational analysis. Thank you.